Hello, welcome back to our series of lectures about action potentials. In this portion of the lectures, we're going to be talking about action potential conduction. Okay, so first, we're going to be addressing this question of how are action potentials propagated down the axon. Then we're going to look at, kind of as a part of that, what is the role of refractory periods in action potential propagation? And finally, what factors affect the speed of action potential conduction? Okay, so let's first start off with this slide, which you've seen before. Hopefully it looks familiar to you. This is the typical shape that we see for an action potential. But the thing that I want to point out to you about this slide is I want to remind you that this waveform that you're seeing here is the changes of the membrane potential at a single location on the axon, right? So what we're doing is we are recording from this one location on the axon, and this shows you what happens to the membrane potential over time at that one location on the axon, okay? However, remember that I told you at the very beginning of this unit on the nervous system, and also just kind of throughout our discussions of action potentials, remember that action potentials travel down the axon. That is not shown here, okay? So we have yet to address this question. How do action potentials travel down the axon? So let's talk about that. First of all, I want to start by introducing these terms. So when an action potential travels down an axon, we refer to that as conduction or propagation, okay? So those are the words that we use to describe that action, okay? Let's take a look at what happens, how the action potential travels down the axon. Remember that I told you that an action potential starts when you have a depolarizing stimulus. In this diagram, we have a depolarizing stimulus, which is a graded potential, coming to this part of the axon, which we refer to as the trigger zone, or at least your book calls it that. In a lot of other contexts, it's called the axon hillock. But I'm just gonna stick with what the book says. Okay, so the trigger zone, okay. Trigger zone is special in that it has a very high concentration of voltage-gated sodium channels. When we have a depolarizing stimulus come to this area that is chock full of voltage-gated sodium channels, what's going to happen? See if you can answer that question. Okay, so hopefully this wasn't too hard for you. When we depolarize the voltage-gated sodium channels, they will open. Okay, so that is what is shown here in this text right here. And sodium is going to enter the axon. Okay, when sodium enters the axon, what's that going to do to the membrane potential? So just answer that question really quickly. Pause if you need to. Okay, hopefully that was also a fairly simple question for you to answer. When the sodium comes in, it's going to depolarize the membrane, right? So here in this figure, sodium is coming in here. It's going to depolarize this area of the membrane here. It's going to depolarize this area of the membrane here. These little arrows show you kind of the flow of the sodium ions, but I wouldn't worry too much about that. All right. When we depolarize those two areas of membrane, right? Remember, we have sodium coming in here, we're depolarizing here, we're depolarizing here. The effect of that depolarization on the axon is actually different in those two regions. Okay, if we look at this region downstream of where the sodium has come in, we've depolarized that region of the axon. That's actually right here. Okay, we've depolarized that region of the axon. What is depolarization going to do to the voltage-gated sodium channels in that area? it should open those voltage-gated sodium channels, right? Once those voltage-gated sodium channels open, we're going to have more sodium coming into the axon. That's going to depolarize the next section of membrane. That's going to open more voltage-gated sodium channels. We're going to have more sodium come in, and so on down the line. Okay, so that's how we get action potentials happening at each part of the axon. Okay, a couple things I want to say about this. One, this is why the action potential doesn't degrade over distance. So remember when we talked about graded potentials versus action potentials, we said that graded potentials, their size degrades over distance. For action potentials, that's not the case. They maintain their same size over distance. And the reason for that is because we're constantly regenerating it as that sodium moves in and depolarizes the next patch of axon and triggers a new action potential, okay? Second thing that I want to say about this is you might have noticed this is a feedback loop. What kind of feedback loop is it? It's a positive feedback loop, right? So sodium's coming in, we're triggering depolarization, triggering more opening of sodium channels. That process is repeating itself over and over again. We're reinforcing the stimulus. That is a positive feedback loop, okay? Okay, so now I've described how we can get an action potential in one area to trigger action potentials further down the axon, okay? Now let's take a look at what is going on upstream in the area that's closer to the cell body, okay? Remember that we had sodium coming in. It's going to depolarize both downstream and upstream. However, that depolarization that's upstream, okay, over here, is not going to trigger an action potential. And the reason for that is because this region of the axon has just gone through an action potential and it is in its refractory period. If it's in its refractory period, it's not going to be able to undergo another action potential, okay? So that is why we have the action potential traveling in one direction. That is why we have the action potential not conducting backwards, okay? Right, I already answered that question and I just went over that. So we'll move on. Okay. All right, so this figure is a snapshot of one axon at one point in time. And what it's meant to illustrate is that different parts of the axon are going through different phases of the action potential at the same point in time, right? So if we were to record from all 11 of these locations on an axon, which is actually physically impossible, we would see that when this region of the axon is just finishing or almost finishing its action potential, okay, the red is what has already happened and the dotted line is what is going to happen, right? So here we're just finishing our action potential. However, if we compare that to other points along the axon, you can see like here at 5, we're just kind of in the middle of our action potential. At location 11, we are just starting our action potential. Okay. So you can kind of compare this to like a wave at a baseball stadium. So imagine you have a wave, a wave where people stand up and sit down, right? So if you have a wave starting on the right end of the stadium and then moving to the left end of the stadium, let's say you take a picture of the whole stadium when the people on the right side have already stood up and are just starting to sit down. Okay, at that same point in time, right, in that same picture, you should see like kind of in the middle of the stadium, people are maybe in the middle of standing up. On the left side of the stadium, people are just starting to stand up, right? So we have the wave traveling down the whole stadium the same way the action potential is traveling down the axon.